minus 50 seconds. T minus 40 seconds. T minus 30 seconds. T minus 20 seconds. Ten, 9, 8, 7, 6, 5, 4, 3, 2, 1, 0.
Good morning, everybody. My name is Martin Leahy. Happy Friday. I want to welcome you to TechFest, brought to you by IT at Cork and SkillNet. Over the past few days, we've explored a lot of different aspects of technology and how it's influencing our lives today. Working remotely from the office, the future of work, data, cybersecurity, and this morning we're going to focus on a more specific aspect of technology as it applies, especially in Ireland today, in the biopharma industry. The biopharma industry is a significant engine in the Irish economy and a significant deliverer of therapy to patients worldwide. And we're going to take some time to take a look maybe a little bit more at the interior of what goes on in the industry, specifically around the area of data and data analytics. We've all spoken, at length about data and what data can do for us, and this is an opportunity to see an industry that has really taken big steps. Data has been a key factor in pharmaceutical and biopharma for quite some time, and it'll be interesting, I think, to see what some of the more advanced projects have done in this area. I'm going to hand you over now to Dr. Maria de Souza Gallagher, who's going to be your compere for the day. And we'll step away from the VE studio here on the Mallow Road and over to the live conference.
I am a senior lecturer, PI, and director of the Masters in Pharmaceutical and Biopharmaceutical Engineering at University College Cork. And I'm going to be your MC for the digitalization of the biopharmaceutical manufacturing track this morning. And if you have any comments, please do them at #TechCork20. So, well, the world is looking at the biopharma industry for solutions. And the industry faces constant challenges in producing drugs in appropriate quality and quantity to keep patients' supply, while also looking ahead to new therapies and vaccines. How fast the industry can respond will shape the outcome of the pandemic and also the reputation of the biopharma industry for decades to come. Cork and the southwest region of Ireland has become a unique ecosystem for driving IT and biopharma clusters. The area is actually home to multinational operations for the 7 of the top 10 global pharmaceutical companies and employing approximately 15,000 people. These local operations have developed deep expertise in bioprocessing and supporting operational technology with many global centers of excellence operating in this region. An equally strong location for ICT and global business services, the region is a hub for over 60 multinational information technology companies, employing around 30,000 people in key areas such as the data and cybersecurity. This depth of both operational technology and informational technology knowledge and the potential for deep collaboration is creating a unique ecosystem for creating fully digitalized biopharmaceutical operations. Today, we'll be focusing on the value and the impact of digitalization for manufacturing competitiveness, and ultimately, these impacts on the healthcare and well-being of patients. So this morning, I'm delighted to be in virtual presence of these illustrious speakers and industry panelists. So without any further delay, please let me introduce the keynote speaker of this morning, Dr. Colin Clark from NIBRT. And Colin is a principal investigator at NIBRT. He has a PhD in bioinformatics and specialized in the application of multivariate statistics and machine learning algorithms to high-dimensional data. He is a PI at NIBRT since 2014, and his bioinformatics group is currently focused on further understanding of the CHO cell biological system using next-generation sequencing and advanced computational techniques. So, Colin, please go ahead.
Thanks, Maria. Thanks for the introduction. I'll just try and share my slides. Good morning, everybody. Okay, good morning. Before I start, just to say thanks again to Maria for the introduction. Thanks to the organisers at IT at Cork for giving me the opportunity to speak today, and thanks for everybody for attending the talk and the session. So I'm Colin Clark. I'm a principal investigator at the National Institute for Bioprocessing Research and Training, and I'm also an associate professor at the School of Chemical and Bioprocess Engineering at University College Dublin. So maybe before I start, I'll just give you a quick introduction to who we are at NIBRT. So as Maria said in her introduction, Ireland punches way above its weight globally in terms of biopharmaceutical manufacturing. We've about $10 billion of investment in capital assets. We've 9 out of the top 10 of the world's biopharma companies in Ireland making biopharmaceutical products. And our success really stems from a young, educated workforce, a good track record with regulatory authorities, and really have the expertise here in Ireland to be able to deliver the business objectives and crucially delivering patient supply from our operations in Ireland. And a small part of that is attributable to NIBRT. So if you're not familiar with who we really are unique in terms of what we can offer people for training. So we have a 6,500 square meter facility based on the University College Dublin campus. It's a simulated GMP facility, so it's exactly the same in terms of the building as you'd find in any of the companies producing a biopharmaceutical. It's on a smaller scale, but it is exactly the same. So the equipment is very similar. The behaviors have to be very similar, and it provides a unique training environment. And crucially, it allows people to come out of a classroom and then get practical hands-on experience in a safe environment. So if you make a mistake, it doesn't have implications on a company. So really, you can make your mistakes at NIBRT. It's actually so similar to a biopharma company that somebody once told us that it smelled the same as a biopharma company. And I think that's probably the best validation we could have got. So it really is tremendous. The training team do a fantastic job. We have people coming from companies in Ireland to train at NIBRT. We have people coming from academia at undergraduate and postgraduate level, and then people coming internationally. So amazing facility, amazing team, and really that's played a key role really in how the industry has grown. We also have contract research. So if you're a company that wants to do some kind of research in making a biopharmaceutical. Maybe you want to characterize your medicine. You can come to NIBRT, do it on a fee-for-service basis, and get results that are of super high quality. I'm based on the academic research side. So one part of NIBRT's mandate is to look that little bit into the future, to see what the challenges are for industry, to take more of a blue skies approach and really push the boundaries of what's possible in the industry. And that's where I am on the academic side. So we kind of have a foot in industry, but also equally a foot in more academic research. So I'm aware that not all of our audience today comes from a biopharmaceutical background. So I just want to give everybody an overview maybe of the differences between what we know are traditional drugs to large molecules or biologic drugs. So most people in the audience, sure, are familiar with aspirin, over-the-counter pain relief for headaches. So if you see on the left-hand side, that's a molecule of aspirin, 21 atoms. If you look at its structure, its chemical structure, it's relatively simple. However, when we look at the biologic, so biologic is a bigger molecule. It's far more complicated. You'll see it has a three-dimensional structure. The three-dimensional structure is really important for its activity. And then that complexity is increased again by the surface of the molecule, which will have sugar patterns. So much bigger molecule, much more complicated, has to have the right three-dimensional structure, has to have the right sugars present on the surface. And in this case, it's a bispecific antibody, so a dual targeting antibody. And this is a protein. So much more complicated and much more difficult to make. And really making that molecule is essentially life and death, because you have to have the confidence in manufacturing to be able to make that molecule every single time in that conformation with that three-dimensional structure, nearly exactly the same. And then have the confidence to give it to a sick person. So really being able to make these molecules is life and death. And if we consider the complexity, an aspirin is really like a bike. And then one of these molecules is like a Formula One car. So you're talking orders of magnitude, greater levels of complexity. Okay, so those are the type of molecules we're talking about when we talk about large molecule or biopharmaceuticals. So really, these molecules are revolutionary, particularly in areas like oncology. So previously, small molecule drugs like aspirin would have been used for oncology. They're not specific. But when we talk about antibodies are like guided missiles. So really targeted, less side effects. And they've improved really the quality of life for about 350 million people around the world. So revolutionary medicine, much more targeted, but again, very, very complicated and very difficult to make. And actually, I think this is probably one of the most complicated manufacturing processes that we have anywhere in the world to be able to do this. And it's amazing what the industry has achieved in a relatively short period of time, about 30 years since people have started to make these biological medicines. Okay, so let's talk about the manufacturing. So molecules is really complicated. It's made in a living system. So in the biopharma industry, we have small factories and we have big factories, okay? And each of these drugs are made in living factories. So where aspirin is made under the rules of chemical and physical systems, so not a lot of variability or heterogeneity, biological molecules are made in living systems. They're actually made in the immortalized cells of this animal, which is a Chinese hamster. So really interesting story about these guys. The cells from a hamster were isolated about 60 years ago. And when people were starting to look at making these biological medicines, the cells of the ovary of one of these hamsters were the best cell line to make these molecules, because they could make molecules in a human-like way. They grew really quickly and they produced a lot of antibodies. So now our challenges are that we need to make one of these highly complex molecules. We need to do it in cells from an obscure animal that have been in culture for 60 years. And then we need to take those cells and we put them into huge reactors. So huge tanks, 20,000 liter vessels, and we grow them for 14 days. And there are more cells in it than the stars in the galaxy that are all producing this protein that's going to be given to a patient. And at the end of that process, the industry needs to be able to verify that protein is correct, and put it into a vial and get it to patients. So it's an amazing industry in terms of what we're able to achieve, and really complicated, and like it is literally life and death to be able to do this manufacturing process right every time. And Ireland, I think it's staggering that Ireland plays such an important role in this industry, and really changing people's lives by producing these medicines. So in my lab, we try to play a small part in pushing our understanding and being able to contribute and impact in a small way to get these drugs to patients in a quicker way, and to help the industry to be able to do that. And really the work in my lab focuses a lot on data, and we focus on data that comes from the small factories, so the cellular factories. We also look at new types of analytics, and then asking questions like, if we had data from every part of the process, what could we do with it and what could we learn? So just some small examples. So we're really interested in single cell analysis, and this is one of, if not the greatest challenge in the industry, is overcoming the inherent variability of those cellular factories to produce that safe and consistent medicine. And with newer types of analytical technology, we were able to go beyond the analytics that we had previously, where we didn't look at individual cells. We just took all cells and grouped them and considered them to be a single sample. We now have new analytical technology, and we've worked with Becton Dickinson in Limerick and in the US to use new technology to capture data from thousands of CHO cells simultaneously in a single experiment. And that's giving us that picture of variability within those cells. And we're using that then to try and look at how the process affects the cells and their production of the medicines. So in the space of 5 years, our data requirements have increased exponentially. In the past, we would have had one of these dots, and now we're capturing about 4,000 cells in each experiment. So we're really getting the level of resolution to understand how those cells are behaving. And again, with that then comes the burden of having so much data and biological data, and then we need to be able to mine those data and then extract knowledge from it. We're also looking at, in NIBRT, at new ways of measuring on bioreactors. So this is a platform that Jonathan Bones, a fellow PI in NIBRT, built. So this is an autonomous system for looking at the quality of the product that's being produced. So this requires no user operation. In the industry, in some companies, it takes about 72 hours to return a sample. In the system that Jonathan and I have put together, this is totally autonomous. You see here, sorry, just get the laser pointer. You see here that this is a bioreactor. It's sitting on this guy, which is this autosampler. The autosampler can draw samples and pass it to a device that measures the environment within the bioreactor, because people in the industry say that the process is the product. So the bioreactor conditions, or the environment in which your producer cells live, that really affects the product. So we're able to measure that bioreactor environment, look at metabolites or small molecules that are in the environment that the cells live. And then really crucially, we have a mass spectrometer, which is able to do automated characterization of the product quality. So things like if the product is sticking together, if it has those right sugar patterns that are important for its effectiveness, and do that all autonomously. Take a sample at a user-defined time interval with no user operation, and then your 72 hours is cut down. So if you have a problem in your bioreactor, you can know really, really quickly that there is a problem, and take corrective steps in your process to be able to correct that. My job in the project is to use machine learning, and I say machine learning with quotes around it. So we've developed a really simple algorithm to be able to allow us to forecast what's going to happen in the bioreactor. So not only do we know now that there's a problem at the time of measurement, we can also look 24 hours into the future to see if there's going to be a problem. And that gives us a time window to be able to make those changes in the bioreactor, or indeed even stop the bioreactor if we have a lot of product in it and we foresee the problem being really serious. Okay, so at this stage I kind of showed you some of the areas we're looking at. So trying to take data from new analytic techniques, and really we see the future of biopharma, and people talk all the time about biopharma 4.0, that we're going to be able to get more and more data. We're going to get more highly resolved data when the instrumentation is being advanced. And we started to ask questions about, if we had all of those data from all of our plants ready at the touch of a button at our fingertips, what could we do? So what kind of scientific questions can you ask? How does this affect your operations? So in this area, there's a school of thought about digitalizing biopharmaceutical manufacturing. And people are talking about factories of the future, talking about things like real-time monitoring. So I've showed you in the previous slide that we now have the analytics to do that. We're looking at fusing it with AI or machine learning, or using multivariate statistics in new ways. And then even looking further into the future, people are talking about robotics and really going automated, more automated in these plants, up into simulated models and digital twins. But the way I think about all of these newer types of technology is that really data lies at the heart of everything. So if you want to have a robot, you need to be able to tell it what to do. You need to be able to acquire data. You need to look at very, very complex data, highly variable data from biological systems, and really have the ability to analyze that data and get the right knowledge from it. So really, in my opinion, the data is the key to all of this. Being able to use data comes from disparate sources, be able to analyze it properly, be able to find the knowledge that you want and act on it appropriately in the light of biological variability. And I think that's the key, and that's what makes it different from all other sectors, is that you have biological systems and your factories are changing, and your factories can become unstable, and to be able to do that is really the key. And when we talk about enablers, these enablers are similar to other sectors within data science. The three cogs: if you're going to work with data, you have to get it out of somewhere, you have to put it somewhere you can work with it, and then ultimately you have to use some way to extract knowledge from that data. So I think that it's very similar in some ways, but the real difference for us is that we're looking at biology. Just, I won't go into this too much, but just to say that a manufacturing process is complicated. There are many steps, in some cases hundreds of steps that you go through. There are many sources of data. There are many sources of variability. Everywhere from raw materials up until your final product, you will be able to get data from it. These processes can be very complex in terms of their organization. There may be parallel pieces of information that are happening over time, and really it's a big challenge to be able to get data from each one of these unit operations and crucially align it, and then, as with many data science projects, make sure that you get to the right point where you have the data you want and it's clean and ready to work with. But these companies produce staggering amounts of information, so this is only going to increase when new analytics come on board. That's experimental analytics, laboratory analytics and instrumentation. So there are many sources of information. Some companies, believe it or not, still use paper processes for some parts of their work. Amgen gave a very good example of the types of data that they produce and the rate of it. So robotic screening, 200,000 data points a day. In manufacturing, 2,000 batch records, 500 million continuous data points per product. And on the source systems, you've got about 5 million data records. And I know there are probably some people in the audience who work in different sectors, and it's probably not comparable to big data that you might get from the likes of Google or Facebook, but really it has levels of complexity that are beyond some aspects of other sectors. And I think that's the important part. So it is a lot of data, and it's highly complicated. And I'm not going to go through this in detail, but I suppose part of our job is to kind of look at all the options that are out there. When we talk about big data technology, we certainly have the three V's. And there are a lot of different solutions out there, some open source, some commercial, some free. And really what we try to do in the group is to evaluate those and see what are the best options for a particular company. So again, as I start to go through these slides, what we try to do is we try to maintain flexibility. So every company is different. Every company has different objectives. Different companies might be producing different products or different processes, and we want to have the flexibility within our research to be able to work with those companies. So in terms of storing our data, you know, we've used things like Hadoop, as many people have. We're certainly not limited to that. And really what I think is the most important part is how you analyze your data. And many people will be familiar with this quote. It's that if you torture your data long enough, it'll tell you. And that's really important in biology. If you're going from fundamental biology up to the types of projects that we work in when it comes to biopharmaceutical manufacturing. You've got a lot of noise, and if you have noise, your models, you know, they might look good when you train them or when you do your in silico tests, but there's a high probability that they're going to fail into the future. So you really need to have a level of conservatism. You really need to bring that kind of scientific aspect to what you're doing with data. And again, within our group, we have, you know, we've skills in machine learning and multivariate statistics. And what we try to do really in this case is to be conservative. So Occam's razor is another one. You know, when all things are equal, the simplest solution is going to be the best one. And really, we don't try and use the most complicated algorithms that we can find, the ones that are in fashion. We find the ones that will work for the data that we have and be able to handle the levels of complexity and the levels of noise, and to be robust when we try to look into the future. Okay, and again, we use lots of different things that everybody else does. And when we prototype, we often prototype rapidly in things like R or Python. Okay, so moving on to the next part of the talk. So when we wanted to get into this area, we were lucky enough to start to talk to Donald Carroll in Siemens. And, you know, I think both of us in NIBRT and Siemens saw a great opportunity in Ireland with the level of industry that we have in the country to start something in Ireland that was going to look at using Siemens technology to push the boundaries of what people were doing in data science. And Siemens have beautiful technology called MindSphere, which is an operating system for the Internet of Things. They have these centers called MACs, so MindSphere application centers, and they exist across energy, finance, traffic management, automotive manufacturing. And really what we wanted to do was collaborate together and develop that system specifically for biopharmaceutical manufacturing, really to set up that digital technology incubator. So NIBRT has this amazing simulated facility, and then we work with Siemens, who have the technology, proven technology, and the experience in engineering to bring that together to develop this system for biopharmaceutical manufacturing. So we started the project about 2 years ago. We've already had industry engagement. So it's one of the first MACs in the world to have industry engagement. And we've worked with 2 partners on very different projects. And really, we're flexible within the project. We want to work with people because I guess from my perspective, I'm working in NIBRT. I don't work for a pharma industry. You constantly have to learn. There are many things that you don't know. And I think that, you know, that's certainly something that's really important to us, is to be able to work with industry, to not try and do things in an abstract way. So the first industry partner, we were looking at root cause analysis to look at a particular problem, very close engagement with the people in the companies, looking at how we acquire data, how we align data, you know, the types of statistics that we use, and then working in that professional way under the CRISP-M framework to be able to deliver that project. And certainly from our side, you know, that was a great experience, because I think, you know, you really learn about the complexity by working with people who do it on a day-to-day basis, and that's their job, to be able to get those drugs to patients. The second partner was a very different project. So in their lab, in their development lab, process development lab, they had many pieces of equipment, but the equipment wasn't connected. So they wanted to take a very complicated instrument and connect it into an online system where we could stream data from it, and then integrate those data with 2 different analytical devices. So very different from the first project where we were using data to hunt for a root cause of a problem. The second project was really about connecting the information, or connecting the instruments that will give you the information. So we were able to work with the company, build a user interface specifically for them, then look at how they're designing their experiments. So to streamline that within their system, so they could cut down the number of experiments that we're doing. Then to go look at simulation to figure out the relationships between variables, and then have all of that streaming to the people who need it at the touch of at their fingertips through the web browser, and then generate the reports automatically. So in one case, you're streamlining their physical work, but also with the generation of reports, that cuts their time down because they can do all the reports automatically. So two very different engagements already with industry. And I suppose the bigger project, and that all feeds into the bigger project, which is the integration of the industrial Internet of Things with data analytics. And that's really where MindSphere comes in. So we have two streams where we're able to work on those specific projects with industry, but then we also have a very active research team integrate the MindSphere system. So in a company, and I mentioned it on an earlier slide, there are many different types of data resource, and we need to be able to connect to those seamlessly. And that's what MindSphere does in one aspect, is that it provides a seamless interface to all of these different databases within a company. And then to be able to deploy Internet of Things assets, to be able to collect data, to be able to manage devices, and then to be able to, if we collect data, to push it into a central resource where we can do something about it. And that's what MindSphere does. And it's proven in other areas. And then we will be able to use this technology in biopharma. So we have a software aspect to MindSphere, but we also have these physical aspects. So if you have a pump or an actuator in your process, within 24 hours, you can connect one of these MindConnect devices to it. And that really reduces the barriers to deployment of IoT technology. It's easy to manage, it's secure, and it can be deployed really, really quickly. And then you can start to connect your plant. And then we hope down the line that we're not just deriving data, but then we'll be able to go back and use Siemens infrastructure then to be able to make changes with pumps or whatever we need to make. So obviously this is more future looking, but the core technology exists in Siemens, and that's the kind of key to be able to bridge the scientific understanding with the algorithms and the statistics and the predictive aspect of it, and then ultimately to be able to have the control. And that's really where a large part of our research focuses. And then we can build our systems, and this is kind of one of the early kind of frameworks that we put in place. So the goal would be to have MindSphere sit as a manager, and it's communicating with all of the resources in your operations at different levels. And then you have your Internet of Things infrastructure. So you're able to connect to your equipment, devices, people, all managed by MindSphere. Then it would connect into a cloud or a local analytics platform that might run on Hadoop or Spark, and then have interfaces like Shiny or Aura, Plotly. And really, this is the first one we put in place for prototyping. But really, we feel the key thing is to get the collaboration between data scientists, subject matter experts in your industry, and then to be able to have all of those data and be working collaboratively, and then being able to deploy your analytics. And it's based on good scientific practice, good data science, and then getting that expertise and collaboration. And this is constantly in evolution. We're using Amazon EMR at the moment to look at new approaches to be able to do this in a better way. So we're flexible in terms of the technology, but in our approach as well, we're able to recognize that new things are coming, and then try to be ahead of that, and then to integrate those within the system. And again, these Spark is a part of that, and really that gives us some of the aspects that we really want in our project, is to be able to do predictive, to stream data as we get it, and from those analytical systems that I showed you near the start, to be able to work with existing resources, and then to have real flexibility in terms of the computer programming languages that we're able to use. And this is really in terms of research, so to be able to prototype really quickly to get solutions. Just an overview of what the system is like. So you can access the system through a web browser, and then you've got the distributed system where you can do analytics, and you've got your data stored in the data lake. Okay, and that's just an overview, and that's a flexible system. And then we can customize the user interface. So we can build these really quickly, prototype them in R, and that's an example of one. But we have many more. And then when we worked with companies, we built bespoke user interfaces specifically for them. And I think that's one of the strengths of the project, to have that flexibility. And the really, really nice thing is that once we get this system to a certain level, then we have a sandbox, both in terms of the analytics system, but also in terms of having a plant. So not to have to go into a GMP manufacturing facility. We can do this in NIBRT within the infrastructure that we have. Okay, so I think I've probably gone a little bit over time, so I'll start to finish up. So in the biopharma industry, utilizing data in a more efficient way is, people in the industry recognize that there's data that's sitting there that could be analyzed in a better way. The industry, of course, over the last few years has got better, as companies like Amgen have led the way, but I still think there's a good bit to go. And it's really, really important that we focus on this, because at the end, the people who benefit the most are patients. And that's what we have to keep in mind all the time when we do this work. It's really important to have collaboration. So as somebody who works in data, the single worst thing that I could do is not talk to people who work on other aspects of biopharma. The processes are so complicated that, certainly for me anyway, I can't understand every part of it. And I learn an awful lot by working from people. And there are great people we worked at in those first two projects that I and the team in NIBRT and Siemens learned an awful lot. We need to be flexible. So these analytics approaches they're developing rapidly all the time. You need to keep up to date with what's coming next. Never get locked into a particular approach. In science, there's always new things happening. There can always be better ways. And definitely getting those IIoT resources and the types of technology that Siemens have integrated within that data analytics project has huge, huge value. Okay, so the last slide, thank loads of people. Peter and Anna in my group who done tremendous amount of work on those projects and getting the system up and running. And then the people in Siemens, Michael O'Donoghue does great work. Siobhan Fleming, who'll be talking on the panel discussion, and was really involved in the business development that plays key roles in our collaborations with companies. Anya Higgins as well for the startup of the project. And then of course, Donal Carroll, who's amazingly supportive from the Siemens side. And really, it's a great team. We all work well together. And that's it for me. And if you have any questions, I'll be happy to take them.
Thank you so much, Colin. I think I am live now. Thank you very much, Colin. Excellent presentation, very comprehensive. And I can see we already have a couple of questions that had popped up on our pads. So if I will pass to you the first question, it's a quite specific one. Hopefully you'll be able to give us some insight. And the question is, can you talk about the digital twin and what are we doing to validate digital data in multiple sources or systems?
So I suppose that we're not building a digital twin, but I think like digital twin is one of those phrases that's it's become very popular over the last few years. So we haven't worked on digital twins yet, but to me, digital twins are in some ways a fusion between classical engineering principles and models and data analytics. So we're not working on it, but as the question asks, yes, validating a digital twin in multiple sources is going to be one of the main challenges. And in terms of tech transfer, you can see like with these biological processes that's a key challenge, to be able to do it on multiple sources and multiple processes. And I think that's something the industry is definitely going to have to focus on.
Thank you. I know you had talked a lot about multiple complexity, multiplicity of data. I think the next question is about, is data bias a challenge in biopharma? And if yes, how do you overcome it?
So I'm not clear on what bias means in terms of the data. If it's the type of data, I don't think it's biased. I think from an understanding, you're always going to be biased because the most variable part of your process is more than likely going to be your bioreactor. And you're always kind of biased to look at, you know, the upstream first, because, or the bioreactor, cell culture, growing the cells part first, where the product is made. But bias is, yeah, it's a very good question. And I think bias is definitely one of the things we want to avoid. And I think, yeah, it's a very good question. You don't want to be biased no matter what you do. And I guess the way you avoid bias is you keep pushing the physical instrumentation to get more data, and that's a really good way of avoiding a bias. So just to mention again, the system we built in NIBRT, or Jonathan Bones in NIBRT built with mass spectrometry online, so to be able to continuously measure your product, that's a way of reducing bias. Because the way some companies do it is they don't collect the product quality until, you know, later in the process. The process, it could be 3 days later, 72 hours later by the time they get the quality data. And that's biased because you don't get that more real-time feedback on how your process is performing. But if you push the analytics, the instrumentation, the ability to measure the product faster, then you avoid that bias because you're taking your data at intervals and you really see the performance of the process. And time is really important because your process changes. So your early process, the cells, you know, they might be growing very quickly, but then they stop growing. So that process is really time resolved as well. And it's really important to be able to see those data as the process continues.
Thank you very much. I think it's an excellent answer. Colin, we have one more question. I don't know if you want to answer it now or we can leave it later on for the panel.
I know absolutely nothing about cybersecurity.
Okay.
So this is one of, like, I freely admit I'm not a cybersecurity person, but I think this is a really nice part of our collaboration with Siemens, is that I don't have to worry about that stuff that I don't know, and it's, you know, Siemens have excellence when it comes to aspects like cybersecurity. So it really draws in different strengths that maybe we have in NIBRT from the statistics or scientific, and that's where really that strength of the collaboration comes from.
Yeah, I suppose later on we can look at that in terms of the complex, how you protect this complex data and how you prevent any potential cyber security aspects that could damage its reputation. I think at this stage, I'd like to start by introducing our panelists. So if I will ask people to turn on their cameras. So we have, I'm delighted to introduce you to Enda Cummins from Biomarin, Siobhan Fleming from Siemens, and obviously Colin, you stay with us, please. And I would ask if you could just introduce yourselves and tell us a little bit more before we go into the discussions.
My name is Enda Cummins. I'm a senior processing engineer with Biomarin. I've been working in the biopharmaceutical industry for over 15 years. And I was working with Colin and Siobhan on a collaboration that I'm going to talk about shortly.
Siobhan?
Hi everyone, my name is Siobhan Fleming, and as Colin has mentioned, I work with Siemens Digital Industries. So my role is as collaboration manager, specifically with the Biomac team, working between NIBRT and with our industry partners, providing advanced data analytics to solve problems in the bioprocessing industry.
Okay, and you have already heard about Colin. So without any delay, I was probably going to move into Enda's presentation. Enda is going to be giving us a case study.
Yeah, is my audio okay?
Yeah, it is okay.
Perfect. So I'll just share my screen.
Thank you.
So, as I mentioned, I worked in partnership with Siemens and NIBRT on a case study for the application of big data and advanced statistical approach to process understanding. So, I'm going to run through the project, the background and the project, an overview of the work that was completed, how we approached the project, and then the benefits, the output, and the learnings that we got. So, as mentioned, it was a collaborative effort. So, Biopharma partnered with Siemens and NIBRT. Siemens, at a very high level, brought the technology, and NIBRT brought the science. So, they had an existing platform called the Biomac, and that matched exactly our business needs. So, the goal was to consolidate and to contextualize data from multiple different sources, analyze, report, and display that data in a user-friendly format, with the overall goal of being able to quickly access data to facilitate data-driven decisions. On presenter view, I don't know what to do about that, sorry. Okay. So the first obvious question is why did we go through this exercise? And one of the main drivers really is that complex investigations happen. Sorry, my thing's playing up a little bit here. So when these investigations happen, they need to be done quickly and efficiently with minimum disruption to the business. So in a typical the typical biopharmaceutical data archives and analytical tools don't always lend themselves to this. So for investigations that are of low to medium complexity, where you were focused on maybe one unit operation or data from one or two sources, the traditional Six Sigma approach works perfectly well. So for a Six Sigma approach, you're going to have a variable or an output Y, which is your variable of interest, and that's going to be a function of a number of different inputs, or your X's. Now, very early on in the process, what you're going to do is do a screening exercise where you reduce down the many variables into the specific variables of interest. So the trivial many reduced down to the vital few. So straight away, you've narrowed the breadth of your investigation into just a small number of inputs. Unfortunately, that's not always possible, and shortly I'm going to give a hypothetical illustration of why it's not possible. So talking about the data acquisition and connection. So the first thing that you find when you do one of these investigations is that the data is there. That's not a problem. There's lots of data, but that data is disconnected. So you need SMEs with domain knowledge who are able to interrogate the various systems and retrieve the required information. And of course, that's going to be an iterative process as the investigation progresses. And from experience, it takes a lot of different SMEs, a lot of time, and as a consequence, costs a lot of money. When you have the data consolidated, you're still left with the job of cleaning it and analyzing it. And depending on your company's position on the data science journey, you may or may not have people with the necessary expertise to perform this analysis. And if you do, you're still left with, well, can you free them up to do this job? So to contextualize what I'm saying and give a little flavor of the complexity and of the processes and the volume of data associated with it, I'm going to run through a complex but not atypical biologics process. So for this example, I want you to imagine that the critical quality attribute that you're interested in is only measured in your bulk drug substance. And that's at the very end of your process. So the first thing you need to do is know what feeds into that bulk drug substance. So there's a number of individual processing steps. So you've got chromatography, ultrafiltration, diafiltration, viral inactivation, just typical biologic processing steps. So for each one of these, you're going to have multiple process, manufacturing, and lab results, as well as a suite of real-time process parameter measurements associated with it. And so far, we've been looking at a one-to-one relationship, and that's fine.
But when the genealogy tracing becomes more complex, is what if you've multiple different, we'll say, for example, UFDF states feeding into the subsequent protein A chromatography step. Now, if you imagine that instead of running a batch process, you're running a perfusion process, and that perfusion process might be running for 100 days with any number of bioreactors. So for this example, we'll say 4. And each of those 4 bioreactors are going to be at different stages of their processing life cycle. So one maybe on day 50, another on day 10, another on day 90, etc. So at any given point in time, all these bioreactors are going to be producing material and filling up harvest bags, which is going to be forward processed. And the forward processing is going to be first in, first out, but there's probably going to be criteria associated with that. So there may be some segregation of certain bags of material, and then the other material is going to be forward processed into the next unit operation, which is going to be blended with 2 to 3 other unit operations, and then processed onto the subsequent unit operation. So again, remember that the critical quality attribute that we're interested in is measured at the end of the process, and this overall process might take 130 days. And the parameter that may be impacting the critical quality attribute that you're interested in understanding may be happening in, say, a particular bioreactor at a particular phase of its life cycle. So given the amount of pooling and dilution and the number of measurements associated with the in-process results and the control parameters, I'm probably not understating it to say that you'd be doing very, very well to make that link between what is impacting that critical quality attribute and tracing it all the way back 130 days to a particular bioreactor at a particular point in time. So now with that overview out of the way, just to kind of talk a little bit about the project that we worked on with Siemens and NIBRT and the steps that we took to go through it. So the first thing we did was we had a workshop where the problem statement and the project scope was defined and agreed, and we identified the information and the data that would be required. Then we took samples of each of those data sets to understand exactly what's involved in cleaning them up, consolidating them, and to get a good understanding of how they all fit together. Next, we took the required exports from all our various data repositories, provided them to the Biomac team, who cleaned them up and consolidated them in a custom app. Then it got to the really interesting part, where in addition to the visualization tools, some advanced modeling and statistical analysis was completed. Finally, there was an evaluation of the models, and a report was generated based on the project goals. So this took about 5 months to complete, and of course it wasn't, a linear process like is shown on the right. It looked a little more like what's displayed there on the left. So just to summarize quickly what I went through in the last slide, is what we did is we took the various data sets from our various repositories, provided them to the Biomac team. They were put in a staging area, cleaned, consolidated, contextualized, then put into a process data store and uploaded to an app that we had access to. And as Colin showed earlier, there's some really nice visualization tools that were in that app. And any user like myself could go in and interrogate the app, variable A against variable B, apply filters, and look at relationships that exist within the process. So what was the output? So we now have a customized app where all the selected data is consolidated, collated, contextualized for on-demand visualization and analysis. So this has empowered the users to be able to go in and assess these relationships across multiple batches and also between 2 different sites. This has also increased our efficiency in meetings, where we were able to assess trends, theories, in real time. So rather than if somebody has an idea about maybe this is what's impacting Y, then we can just pull that up live at the time and assess the relationship there. So the other part of the project, we wanted to look at one particular critical quality attribute and determine what factors impact that. So the team used a number of analytical techniques, both unsupervised k-means clustering and principal component analysis, and supervised random forest. And both of those actually correlated very well in what they identified as the variables of interest. So now we've the static database available to us with the historical data from two sites. And one of the next steps is to see if we want to convert it into a live database. One other benefit is that the framework is built and the scripts are written. So if we ever do need to kick off a very complex investigation, the base and the building blocks are there. So then just in terms of the learning, so like for me, like approaching this as a collaboration effort with a defined goal and scope was a massive advantage. So there was a massive amount of efficiencies from working with the Biomap team. This type of project is where their expertise lies. It would be difficult to identify a small number of people within an organization that have the necessary skill sets in data structure, statistics, and the scientific understanding to be able to undertake this exercise. So again, it just made excellent business sense to go down the partnership route. Another thing that jumped out is how well suited the biopharmaceutical industry is for this type of service due to the volume of data and also the complexity of the processing interactions. Finally, it emphasized the competitive advantage that there is in having a workforce that are data literate and able to use basic statistics like that are available within this app that has been created in order to be able to better understand the process. And some of the other things that we'll follow on with in terms of exploring and looking for opportunities is, as I mentioned, converting this into a live database, assessing how we would integrate this app into our ongoing process monitoring strategy, seeing if we can automate elements of our CPV reporting, and then seeing how it can be applied to other areas of the business. So for example, connected labs, process development where they're doing DOE and characterization studies, and then also how we can retrieve data from our contract manufacturers. So that's it from me. Thanks.
Thank you very much, Enda, for excellent presentation. Very detailed about what are the challenges that industry faces in terms of gathering information and bringing in knowledge in order to develop some of these complex drugs. I was wondering, rather than asking you any questions at this stage, and as you mentioned, this is a partnership effort, a collaborative work. Maybe if I could bring back all the people in the panel, so Siobhan, Colin, because we can start the discussion, but also I have already one question that was asked, but maybe there is other things. I think some of the, in fact, that question have been probably answered with some of the detailed knowledge that Enda has provided in his presentation. And this question was for Colin, but we can look at that. Considering what we are dealing with, Colin, I don't know if you would like to keep again the ball rolling. Is we are dealing with very complex molecules that are produced in a complex manner, and with that process, we need to obtain data that will relate to some of the key important aspects of that product in terms of quality, in terms of safety, in terms of what is the target objective of that particular product. So I leave this up to the floor now to maybe discuss this a bit better. What have been the challenges in terms of producing, ensuring this quality and this safety that will deliver safe drugs to the patients?
Are you talking specifically, Maria, about the challenges in drug manufacturing or the challenges around data analytics for bioprocessing?
I was talking it in a whole, so it, very much what I was trying to, as Enda finished his presentation, this is a partnership effort. There is lots of different different types of knowledge that are necessary to ensure that this product is going to be produced of that high level and of acceptability. So we have from life science point of view, from an engineering point of view, from the data analytics point of view, but as we heard as well, is how we are going to collect this multiplicity of data, how we are going to harness, and how we are going even to ensure its security. So as you can see, it's a very big and complex system, and that's we have the right people in the panel to tackle all these aspects of this supply chain.
So, Maria.
Sorry, go ahead.
Okay, I was just going to say, actually, Colin, that you did a great job explaining the complexity of the actual process, and particularly like comparing an aspirin to a monoclonal antibody, and I suppose the variability that can be within that. So there's probably at least two main things that I would see there. One is that you only get one shot at it. So if you're making an aspirin, you can do some rework and reprocessing. And as Colin mentioned as well, like the process is your product. So like you're relying on everything going 100% right all the time. And as I mentioned, you can have batch reactions, but you can also have perfusion reactions that are going on for 100 days plus. So getting that consistency and that repeatability is a massive thing, and we try to ensure that by taking lots and lots of data. So we would have all of our instruments reading back to Pi, and they're taking real-time readings and they're stored in a data archive. And we're also taking on-the-floor readings and they're getting stored in a primer. Other results are getting stored in LIMS. We've Oracle to trace the batch genealogy. We're introducing, we'll say, MES to kind of record various things that happen on the floor in terms of batch records. But what you see happening quite quickly there is you have a lot of different repositories, and it's difficult to have the right people available to get to know exactly what information they want when they're doing an investigation. And if you have that one person, the likelihood of that person being an expert in the various data repositories and being able to efficiently retrieve the necessary information, that's quite low. Then when that's retrieved, there's going to be an element of cleaning it. And then you're also going to need to be able to do some statistical analysis. So you're really looking for one hell of a bioprocess specialist type person there. So if some of the labor is already built into an app, in the sense that, okay, you've got your processing expert who knows what they're looking for, and they can go to the app and retrieve pre-cleaned, pre-contextualized data with some of the basic statistical analysis already built into the app, then you're in a much better place to be able to react to investigations quickly and efficiently.
Thank you. And I don't know, Colin, if you would like to follow up from that. I mean, one of the questions we had before was, in order to analyze a system, it's key to get good data analytics, and how how does this relate to, how are you obtaining this data in biopharma, and is there gaps that in data to make good to make these analytical models? You had mentioned before that some of the data could be obtained obviously from the bioreactor, obviously where is the product being produced. So all those conditions, all these processing factors can affect how this product is going to be expressed and produced there. And then we have the whole downstream process where the product is going to be purified before it would be ready for delivery to the patient. I don't know if you want to mention or Siobhan, if you want to mention.
Yeah, I can talk a little bit about that. So I think that the answer is that we will never have enough data. So we will never understand everything, because the process is really, really complicated in terms that you have living cells and you make a product, and then that product goes downstream. We never have all the analytics that we need. If you look at the cell, and I'm kind of biased towards looking at the cell. So over the last 10 years, we've taken this quantum leap and been able to understand cells in terms of their molecular structure, which is really important because you're producing a product in a cell, but you also need the machinery of that animal cell to be correct in order to synthesize the protein, to be able to fold it, to be able to add the sugar patterns to it. So in terms of, we will always have gaps in the analytics of those cells. We've taken huge leaps in terms of being able to look at its genome more accurately. That's really important because your foreign or your recombinant product is a DNA sequence that's inserted into those cells. So to be able to look at that, so you really go from the finest molecular details of those cells right through to the filling process. And you really need, will we ever have all the data? Probably not. Will we have enough data? I think we'll get there. We're getting there. We're getting closer. I mean, for recombinant proteins, we can already get a lot of data. So in terms of, like, just to give maybe one example of how the cells are connected to the vials at the end is around host cell proteins. So your product, your monoclonal antibody is a protein, but the CHO cells also have proteins, and we call them host cell proteins. And these are very, very important in terms of a critical quality attribute, because if your host cell protein goes through your process and then somehow ends up in a vial, that can cause an immune reaction in a patient, and it can be very, very dangerous. So companies want to be able to determine the levels of host cell protein, and specifically which proteins from your cell factory end up further downstream in the process. So we need to be able to identify those, and a protein is a chain of amino acids. But if we don't know what the sequences are of the amino acids that come from your producer cell, then we won't be able to have the analytics to be able to detect those. So in terms of that, that's one area the analytics can be pushed. What are called metabolites in the cell culture are also another area that you need to be pushed further, because the identification of specific metabolites, these small molecules in the environment, is also very, very difficult. So we definitely need data to build good models, but to do that, we need to push the instrumentation further and further and further. And that's where really we can get more and more data. And that's pushing the instrumentation in terms of the level of resolution of what it can see, but also how we use it. So to be able to have those aspects like auto sampling in the reactor or at the various stages of chromatography in the downstream are also important. So time is a factor also. So we need to push the analytics are constantly being pushed. And just to talk again about NIBRT. So Jonathan Bones, who I mentioned, has a collaboration with Thermo Fisher Scientific, and he's really pushing the analytics, the characterization of the proteins, even now to looking at the monoclonal antibodies that are intact, so not broken up as they traditionally were, but looking at the intact molecule. And then of course using those auto-sampler systems to give you that dimension of time to be able to sample from the reactor really, really quickly. So those are kind of where I would see the instrumentation being pushed. And really for the data analytics side, all our work is based on the quality and the types of data that we get. And the more of it, the better. And the different dimensions that you can get, and then integrating those dimensions is where you'll get the value, where you'll be able to create really accurate models.
Colin, I think you said one word that I could see as a stepping point for Siobhan there. I mean, we can all have we all wish for a lot of information, a lot of data, but sometimes this data can be quite complex as well. But it's how we integrate it and how we relate it to the outputs that we are trying to produce is obviously key. So I don't know, Siobhan, would you like to tell us a little bit more? How do you have been doing that in Siemens?
So I think following on from points that both Colin and Enda made, Enda talked about, different specializations within the biopharmaceutical plant and across the value chain, and Colin then talked about, a lot of data is there and we need to get it together. I think one of the key challenges that we have is the existence of data silos throughout the bioprocess, whether that's at the R&D laboratory stage in manufacturing, or eventually now as we look forward to incorporating patient data. So plants in general are very highly automated, and as you said, the data that they generate is very complex and has huge potential value through data analytics, but it's sitting in disparate silos. So Colin showed us a list of those earlier, like your ERP systems, process control, manufacturing execution, LIMS, and so on. So it can be very difficult to consolidate them. So what we do is we work with standard data communication protocols. We have teams of connectivity and cloud computing experts, and then people who can cleanse and contextualize that data. Somebody mentioned data gaps, so it's about identifying those gaps then and figuring out whether we need to fill those gaps, or is that data that perhaps needs to be eliminated from the data set because it's going to skew the results because of missing data points. As Colin said, we'll never have enough. I think another big challenge is having computing systems and computing capabilities that are capable of handling that data and processing that data, no matter how much we throw at it. So Enda mentioned, the complexity and the volumes of the data. So we talk in big data about the 3 V's, the volume, the velocity, and the varieties. And I think that's more true in bioprocessing than in many other industries. So using the big data technology to ensure that you can manage the data that's coming at you now, but that you'll also be able to manage the data that's coming in the future as we look towards the Internet of Things, connected devices and sensors, and the ever increasing volumes of data that are going to come our way. The business information systems that departments are using at the moment have limitations, and you really need to be building a framework now that will allow you to consolidate all of that data and also move away from the data silo mentality, which is partly connected with the functional divisions within plants, so that you can share data across functions and collaborate through things like visualization, which help people to understand the other perspective in a more straightforward way. I don't know if that answers your question.
It did perfectly. I think it also links to, we had some questions from the audience, and I think the way you have described it is that we cannot obviously operate in terms of silos. We need one of the questions is how we coordinate the different resources from a project management perspective. And I think what you were just saying is we need information coming from different parts of the company, and also that this information will be integrated as the product is being developed from the research all the way into until the quality of analysis of that product. I don't know, Enda, if you would like to elaborate a bit more on that.
Sure. And just on the previous question as well, I think that like, you know, what Colin and Siobhan were saying is bang on. Like, you know, we're always going to want more data, and I think a lot of what we do still is quite manual in terms of taking samples from bioreactors, bringing them down to the lab, getting them tested, you know, all the controls, documentation that's associated with that. It's very labor intensive, and absolutely where we want to get to is the kind of automated sampling and PAT technology that Colin was talking about. But I think as an industry, we still have massive amounts of data, and what we're doing with it now, we could probably do more. So like we have these silos of data, and first of all, as mentioned, they're disparate. So that's what we were doing as part of the Biomac project, is bringing them all together. But a lot of, like, all companies are going to have this data, but I don't know how many companies would have taken the time to do, we'll say, the random forest or any of the analytical techniques that Colin has kind of, Colin and the team have applied in order to gain those insights. So to me, it's kind of, there's probably two aspects to it, but they can happen in parallel. So we can look at getting more data, but I think first we should look at better using the data we have to gain further insights. So usually that's done upfront in process development, but there's nothing stopping us finding out more about our at-scale manufacturing processes.
Mm-hmm. Thank you. And we have been talking about silos of data, but yeah, the data is operated by people, and we can also consider that could be silos of people. At the end of the day, we want the information from different parts of the manufacturing operation will be integrated, is how people work together. And this brings me to some of the other questions we had. Is what kind of skills do people need to have to operate in a process like this? We have, as I mentioned, we have heard the knowledge of the life sciences in order how to transform some of these molecules, how are these cells into producing these specific and complex molecules, how we are going to grow them, what factors influence that growth. So we can look how we are going to scale up this process. So quite a lot of engineering where the data analysis can come at all these points of the way and should come up front when the research and development process is taking place, all the way down to all the quality analysis points that could be throughout the process, so that all these information could be integrated. So one of the questions that we were having is what skills do people in this sector need to have that could conciliate all these type of disciplines. And sometimes we can even say we cannot not know all, and that's completely true, but at the same time we need to understand each other, and we have to be open to have the vision to see where each person is coming from, so that the sum of all the parts is bigger than each one of the individual aspects. So I don't know if any of you would like to come up and tell us a bit more what has been your journey in terms of what needs you have envisaged that people working in this sector should have.
I think it's probably it looks like a two-part question. So one on the biopharma space maybe might be a question for Enda.
Yeah,
I think
sure.
on that side, and then probably one about biopharma and data, which I'll maybe answer after that.
Yeah,
sure.
Just before, before I was going to that, like there was a question before in terms of what are the skill sets that are required to work in the biopharma industry. And I think as I mentioned earlier, we have a particular ecosystem here in Cork. We have two major institutions, University College Cork, also Cork Institute of Technology, that obviously are providing all the graduates that industry is requiring, because sometimes we can't forget, we can have all the machinery, all the equipment, but we need people. We need people with the right skills, with the right mindset, with the right management skills to integrate and deliver the particular molecules, the manufacturing, and ensuring that this is being produced at very high standards as it is required with most of these biopharmaceutical products. And in order to accomplish that, obviously the digitalization is bringing a huge advantage in how we can measure what is happening along that process, anticipating issues so that they could be minimized as they are being happening through those processes.
So I'll take the first half of that question, Maria. So I suppose like biopharmaceutical production is a multifaceted industry. So like obviously you're going to need people with engineering backgrounds, process engineering, mechanical, electrical, then automation is a massive part now and becoming bigger. Then you're going to need people with a deep scientific understanding, and particularly in the area of biologics. You're going to need people that work like hands-on on the floor, who may have done a course that I saw mentioned there in the comments earlier. It's ran in CIT, and I suppose there's many avenues into the biopharmaceutical industry. Like again, it's going to need accountants, project managers, and now more so the likes of data science, which probably wasn't as big a part of the industry, 5 or 10 years ago. So yeah, I think there's lots of ways in, and I suppose like it's getting back to the theme, no one person is going to know everything. So you're going to need people with very specific niche skills, and then you're going to need people who have a breadth of experience and know what people to draw on when it makes sense.
Colin, I don't know if you want to come in.
Yes. So my educational journey was what data science skills do people need, or how do you become a data scientist in biopharma? So I began as a wet lab biologist, so applied biology, and then got an interest in computer science, and then did a master's in bioinformatics and a PhD in it. And I suppose that's what I'd like to say is that, data science in biopharma is different because it's really important to understand your data, which is kind of an obvious thing to say, but when it comes to looking at scientific data, none of the ways of generating data are perfect. So there will always be noise, there will always be variability. And I think it's really important from a data perspective to understand the underlying fundamentals of where the data comes from and what's the performance of the assay, to understand, what kind of tools from the data science world are applicable for those types of data, because there are many, many very complicated types of statistics out there, highly, highly complicated, that were probably not developed for biology when they were designed. And I think one of the things I would say is that those two parts of your education for data science and biopharma are really important. So to know what your data is, and then to know the limits of what you can do with it. So not to use something that's really, really complicated, that might even be a black box model to go and start analyzing data, because it's probably wasn't designed for it. And unless you understand both sides of what you're doing, there can be pitfalls. And I think that's really important part of the understanding that is really important for people working in this area, is to not use, you know, those kind of statistics on data. It just might be suitable for, and to do that you need the understanding of the science as well as data science.
And just to echo that, actually, Colin, that was one of the massive benefits that we got from working with the Biomac team, because I think if we had just got in an external data scientist who wouldn't have had the background in, you know, the biopharmaceutical processes, you probably come up with a lot of very strong correlations which are technically correct but not really relevant because they're known. So that was a massive advantage in partnering with Siemens and NIBRT.
So if I jump on that to Siobhan, obviously we can produce things, we can get a lot of data, but if we don't have a user-friendly platform that can integrate and helps the decision-making process, maybe we'll end up nowhere. So could you tell us a little bit more about the Biomac, and or how you can come into this partnership and facilitate the connectivity between the life science people, the engineering, the data analytics people, and make each other's jobs easier and accomplishable?
Yeah, so I suppose the purpose of Siemens working with NIBRT, Siemens obviously has existed for over 170 years and has a, you know, very strong reputation for providing industry solutions. Within the pharma division of Siemens, we have a very strong focus on the customer and on the patient. And the intention at all times is to understand customer challenges and then address those challenges through technology. So with Biomac, the problem that we identified was disconnected data, to put it very simply, and then to use that data to address problems, to identify where processing could be improved, to increase efficiency, or to achieve process intensification. And then Colin touched on it earlier, to eventually move towards what we've called disruptive process control, where you can then monitor those parameters in real time through your process, and eventually use machine learning and artificial intelligence to allow the system to correct those parameters and make the adjustments required during the processing, or even to just suggest those to the operators of what changes might need to be made in order to make sure that you're falling within your golden batch parameters on every batch. The fact that it's a big data system that we're working on in Biomac means that we're working towards a place where you will be able to compare your current batch with every previous batch of the product that you've ever made. And again, that's one of the benefits of working on a system that supports larger volumes of data. So the context of the Biomac platform really is to combine the extraordinary technology that's out there at the moment and that's coming down the track in terms of the industrial Internet of Things, cloud connectivity. Somebody mentioned cybersecurity. That's obviously a huge consideration as well. Within Siemens, we have the capabilities to address all of the technology issues. And of course, we also have people on our team as well who are both data scientists and bioprocessing engineers who also understand the science, and work then closely with the NIBRT team, who bring the very deep bioprocessing expertise and can help us then to build out a demonstrator within the NIBRT facility to allow us to show these technologies in use to our customers. So the premise is that you, through communication protocols and connectivity, you incorporate the data from the various systems that you have into the data analytics storage area for contextualization and cleansing and readiness. Colin mentioned removing the noise, so making sure that the data you have is good quality data before you start your analytics in anger. In the Biomac analytics platform, we then work in a very very close and targeted way with our customers to make sure that what we're building for them addresses the specific challenge that they're looking at. So you have challenges right across the value chain. It could be from the R&D lab, which was the project that Colin discussed earlier. It could be a problem in manufacturing, or it could be something in supply chain, trying to understand what the status of your raw materials is. It could be working with contract manufacturing organizations to understand whether they're meeting the targets and the agreements of the SLA that's set out with them. So we need a system that's going to, an ecosystem really, that's going to allow you to incorporate all of that data in the Internet of Things world as the volumes of data come our way. And then you can deploy the analytics that are appropriate to the challenge that you have identified, and really empower your people to make informed decisions and use data-driven decision making. I was speaking with somebody recently about the skills shortage that's coming in the bioprocessing industry, and how, you know, relying on somebody's experience isn't always going to be possible in the future, and it's wonderful now to have the potential to use data to make those decisions in a more certain and informed way.
Thank you very much, Siobhan. We still have 3 questions, and on the tone that you just mentioned there is about people having the right skills, and without repeating myself, I mean, we are very lucky to have such 2 major institutions here in Cork that are in very tight connection with some excellent centers on this area, both on the IT as well as on the biotech side. Both in University College Cork, we have different degrees that are providing graduates with the right skills to tackle all this complexity of systems, and some of them obviously integrate more explicitly or not some of the data analytics and digitalization aspects of the processes, because from some years ago, as Enda was saying, the way we were doing business was very different from the way we need to do business, and obviously those skills have to be integrated in order to prepare and provide the skills of the future. Having looked at 2 other questions we have on the screen, we need people to carry out these processes, and why are we producing some of these drugs? I think I'm going to jump to the last first before I go to Enda for the other question. Is about sometimes this question is for Colin, but sometimes we should not forget that why we are producing biopharmaceutical products is because there is unfortunately patients out there that are suffering from very serious and rare diseases, and we are going through a pandemic stage that obviously had brought us these concerns more and more to our minds. Without, I don't know, Colin, if you want to come in regarding the aspect that when we are manufacturing things, we need to have in mind that we are targeting, or we are not targeting, but we are hoping to deliver a high quality and safety product at the right time.
Yeah, I think that's the most important thing everybody needs to kind of always keep in their mind, that the outcome of this is something that's going to help a patient. And you know, anything we can do in any aspect of biopharma to get those drugs to people in a faster way, time to market, being able to make sure that, you know, cost of goods is appropriate for patients and the company, is any aspect of pushing the technology on. I think even if we look further down the road, you know, we spend a lot of our time talking about recombinant proteins today, but the industry is also rapidly changing in terms of the modalities or types of drugs that are being produced. So we have now the emergence of gene therapies, In the last few years, we've seen approvals in gene therapies and also new cell therapies. So the challenges are still there, and the onus is still on the industry to be able to get those medicines to patients, operating in a very complex, what is now very, very complex environment in terms of supply chain and also as well the manufacturing side.
Mm-hmm. Thank you, Colin. And I don't know if you want to comment on the monetary value of delivered for the work. So what represents for biopharmaceutical industry the value of having high quality on-time products that will deliver the needs of these patients?
So in terms of the value that can be derived from, I suppose, the app and the statistical understanding and having the right information available at the right time, like it can start right from when the process is being developed. So in order to refine and develop your process in the most efficient way, you're going to need to do a number of studies and designs of experiments. And if those can be automated and the data fed back live, and then the interrelationships between the various parameters understood so that the process can be properly understood and characterized and made efficient in terms of the reproducibility and the quality, then that's going to save an awful lot of rework down the line. And then in terms of having, like, to me, part of what we've done with this project is almost like an insurance policy. So if anything throws up to be a problem down the line, we already have the infrastructure and the tools and the partnership there so that we can get done in what might take, you know, multiple months in a couple of weeks.
Thank you very much. And I don't know if anybody has any other comment or question. I think the time has passed, it flew by. And any question, any comment? I think we have answered broadly all the questions that had been put to us. So at this stage, I'd like to thank you all for participating in this very interesting and relevant discussion. And hopefully we'll be all in touch soon so that things can progress further. So for all of us that work or in industry or in collaboration with the biopharmaceutical industry, it has never been more clear that the need to reduce the time to market for life-saving therapies is a race against time. And we have been having really interesting discussions today about this challenge and how we can use digitalization to propel the industry forward in its mission to save lives. And one sentence that often I tend to say even to my students, it's let's not forget, you can only manage what you measure. So let's measure the right things so that we can produce the right things to deliver the products that are in need. So thank you very much for your attention today. And if you have any further comments or questions, you can do that at the hashtag TechCork20. Thank you. Thanks.
Thank you, Maria.
Thanks very much, Maria.